Welcome back, dear students. In the first lecture, we have studied the general chemical composition of the plasma membrane and percentage of the protein, lipid, carbohydrate, basic structure of the plasma membrane, right, along with the video. Now, in the present lecture, we will be studying the basic, that is worldwide accepted model of the plasma membrane called as a fluid mosaic model. But before starting with the fluid mosaic model, one point was left, so that is carbohydrate, right? Percentage of a carbohydrate present in the plasma membrane. So glycolipids, carbohydrate, basically two types of carbohydrates are there in the plasma membrane: glycolipids and glycoproteins on the external surface of the cell, right? Yes, the carbohydrates are present in the plasma membrane in the form of a globular, means a glycoproteins, and the glycolipids. Okay, glycoproteins and glycolipids. Now these glycoproteins and glycolipids present on the external surface, and this oligosaccharide chains plays an important role in the cell-to-cell recognition process. So this oligosaccharide chains of the glycolipid, means a lipid, means a glucose molecule oligosaccharide associated with the lipid, so that is called as a glycolipid. See, written here, glycolipid and the glycoproteins. Why they are important? For what they are maintained on the cell surface? They are useful for the cellular recognition, right? If you touch your skin, right, now you will if your eyes are closed and someone touch your skin or just pin you, then what? You will recognize it with the help of some kind of a sensation. Similarly, cell to cell recognition is there, and that is carried out with the help of this glycolipids and the glycoprotein molecules, right? So list amount, that is only 10% of the glycolipid and glycoprotein, that is carbohydrate, present in the plasma membrane for the cellular recognition. Now let's move ahead and see the basic model of a plasma membrane, that is called as a fluid mosaic model of a plasma membrane. So this fluid mosaic model of a plasma membrane was proposed by the scientist Singer and Nicholson in 1972, right? 1972. So long time back this model was invented by the scientist, and it is widely accepted as compared to other proposed models. Now let me tell you varieties of different types of models were there, like Danieli Dawson's model, as well as many other scientists. Unique membrane model, like the models were proposed by the different scientists, but none of them is accepted, because no doubt by the time they were accepted, right, but as soon as research were conducted and whatever outcome is there, on the basis of that, scientists, they confirm the fluid mosaic model as a applicable, or you can say the basic model of the plasma membrane, actual model of the plasma membrane. That's why the unit membrane model as well as a tri-layer model, these models were outdated right now, and hence they are not included in our syllabus. The one which is accepted worldwide, that is fluid mosaic model, is nowadays included in your syllabus. So why this model is being included as, or it is been having so much importance? So important feature of this model is that the biological membranes are considered to be quasi-fluid, that is half fluid. The biological membrane is considered to be half fluid, right? And this model, that is fluid mosaic model, is going to explain that concept, quasi-fluid. So it's a structure in which the lipid, right, in which the lipids and integral protein molecules are arranged in a mosaic form. Their fixed pattern is not there. It is not like after protein molecules 10 lipid molecules will be there, and after 10 protein will be there, after 10 one more protein will be there, or integral or transmembrane proteins will be at specific location. No, such a fixed pattern is not there. They are arranged, means the protein and lipid molecules, they are arranged in the mosaic form. They are arranged in a random way. The reason for the wide acceptance of this model is that it is very well explained the properties of a cell membrane. Whatever properties of a cell membrane are very well explained by this lipid that is fluid mosaic model. The model assures that there is a peripheral layer of a phospholipid. This peripheral layer of a phospholipids in which the peripheral proteins as well as the integral proteins, right? Peripheral and integral proteins, they have been embedded to a varying degree or with a various percentage. So that's why this model was best and accepted worldwide. Thus, the proteins are compared to icebergs. So, as they are mosaic, or the proteins and lipids, they are mosaic, the random pattern is there, no specific pattern is there. So, this model is going to give us a similarity or yes, resemblance with the icebergs. You might have seen the icebergs, or you can say ice cubes are floating on the ocean or a water surface, right? So that ice cube can go anywhere. Similarly, or iceberg can go anywhere on the ocean or that on the water surface. So similarly, these proteins, they can freely move into the membrane, right? So this is kind of a similarity observed between the icebergs and the lipid bilayer model. So proteins are compared to icebergs floating on the sea of the phospholipids. So they are considered to be the icebergs floating on the sea of the phospholipid bilayer. This is a one layer, this is another layer. Two layers are there together called as a bilayer, right? So during certain immunological experiment, the attachment of the antibodies to the cell surface, as if you remember, just I told you earlier, the glycolipids and glycoproteins and glycolipids are used for the cellular recognition. Whatever molecules are coming across the cell, they are recognized by this glycolipid and glycoprotein. So whatever antibodies are coming across it, they are exposed or they are detected by the proteins such as a glycophorin, which is attached to the cell membrane, right, which is attached to the cell membrane. So this model considered that lipid protein association. What it says, this model considered that the lipid protein association to be hydrophobic. But now the fluid mosaic model, it says that whatever, see, if the protein molecules, they are having different property, this lipid molecules, they are having different property, but as this protein and lipid molecules are together, binded together, so some kind of association must be there, right? Agree with this? Yes, some kind of association must be there. As let me tell you the example. As we are sitting in the class now, right now, not sitting in the class, but just imagine if you are sitting in the class with your friends, some kind of a bonding is there with a close friend, and those people we don't like them, having also some kind of a bonding, right? Is it there? Yes. If the people or if the person is your friend, then you have a good type of a bonding, affinity towards that person. So that can be a hydrophilic. We can correlate the terminology. That can be a hydrophilic because you like the person, right? And another one you don't like. So you try to pull that person away from you. So that can be hydrophobic, right? Hating kind of a water hating, that is hating kind of a association is found. So if the people, or in any case, if two things are residing together, there is some kind of a bonding found between them, right? Isn't it? So in this case also, if the proteins and the lipids with the different biological properties, if they are found together in a plasma membrane, in a lipid bilayer, so there must be some kind of association. That's why they are together. Maybe the hydrophilic or maybe hydrophobic, but some kind of association is there, right? Now, some scientists says that in the lipid bilayer, that is Singer and Nicholson, says that the association takes place relationship between the lipid molecules and the protein molecules is the hydrophobic, right? It is hydrophobic because this core of the plasma membrane, that is lipid barrier, is hydrophobic, and this protein is also having some kind of a hydrophilicity inside it. So hydrophobic association is there, right? Water hating kind of association is there. But the Danelli and Dawson assumes that, another scientist who also put forth one more model for the plasma membrane, they said that it is a hydrophilic. They said it is a hydrophilic because the core of the protein is hydrophilic. So again, questions means ambiguity is there whether exactly it is hydrophilic or hydrophobic, right? Danieli Dawson says it is a hydrophilic one, association between the lipids and the proteins. But this model, fluid mosaic model proposed by the Singer and Nicholson, said that this association, that is between the lipids and the protein molecule, is what? Is the hydrophobic one, right? Fine. So the fluidity of the membrane is a result of what? This hydrophobic interaction according to this model, that is a fluid mosaic model of a plasma membrane. The fluidity, that is flexibility of the plasma membrane due to the hydrophobicity of the or hydrophobic interaction between the lipids and the protein molecule. So this phospholipids and many intrinsic proteins, right? Phospholipids, these are the phospholipid. Entire head, like head and tail, is the phospholipid molecules, and many intrinsic proteins, right, are amphipathic molecules. As I said earlier, phospholipid molecule is also amphipathic, amphoteric, and the protein molecule is also amphipathic. That is both a hydrophilic layer and a hydrophobic group occurs within one and the same molecules, right? Amphipathic means what? Dual in nature. And how the dual in nature? So if you talk about the lipid molecule, so its head is hydrophilic, whereas the tail is hydrophobic. Similarly, the protein molecule is also showing the hydrophilicity and hydrophobicity, right? Okay. So the globular, hence, okay, that's why the protein and lipid, they are considered to be the amphipathic molecules, right? Okay. The globular proteins, see, these proteins, they called as a globular protein. Let me show you in another diagram the name globular proteins. Yes, intrinsic proteins, they called as a globular proteins. Fine. So globular proteins, these are said to be the globular proteins, occurs in the membrane, are of two types. Yes, we studied them: external or extrinsic and intrinsic, right? That is, extrinsic is said to be peripheral, intrinsic, they are also called as a integral proteins, right? Okay, so the peripheral proteins are the soluble and readily, yes, we also discussed it earlier, the peripheral proteins, they are soluble and readily dissociate from the membrane. They are present entirely on the outer side, okay? Whereas the extrinsic proteins, they are closely attached with the plasma membrane and they are not easily dissociate from the membrane. They remain attached with the plasma membrane. Their dissociation is very, very difficult. So some proteins may partially penetrate through the surface, like this, partial penetration is there, while others may penetrate directly through the lipid bilayer facing cell exterior as well as the cell interior. So such a proteins in contact with the aqueous solution, aqueous solvent of both the side, that is from the outer side as well as the inner side of the cell membrane, right? The integral proteins are amphipathic. So those integral proteins which are in direct contact with the aqueous solution of the cell outside as well as the cell inside, they are called as amphipathic molecules, right? And they are having hydrophilic and hydrophobic groups. So integral proteins are having hydrophilic and hydrophobic groups. So they are hydrophilic polar heads. Now here in this case, just like this, the polar heads of the lipid, and here is also hydrophilic polar heads of the protein molecules. right? They protrude from the surface of the membrane, and while non-polar hydrophobic groups are remain embedded, means the core portion is hydrophobic in the interior of the membrane, right, in the interior of the membrane. So the integral proteins, now I am repeating this, hydrophobic portion is penetrated, remain attached with the membrane or penetrated inside the membrane, whereas the hydrophilic portion is found to be protruding outside the membrane on both the sides. So the integral proteins are capable of a lateral diffusion. It doesn't mean they can drop down. No, they are capable of a lateral diffusion. They can diffuse here like this, like this. They can go anywhere in the lipid bilayer, right? Okay, so fluid mosaic model. Now let me show you the video of fluid mosaic model so that it will be very easy for us to understand. So dear students, here is the cell, right? Now I'm going to show you entire things once again. So the cell with the plasma membrane. So as I said earlier, the plasma membrane is what? It is fluid in nature. Now here in this video also you can see that, you can observe that the lipid bilayer, phospholipid molecule, this is one phospholipid molecule, this is another one. Cholesterol are found to be embedded inside it. Glycoproteins are also there. Protein molecules are also there, called as a integral and yes, this is also integral but peripheral protein, right? Let's see further. So you can see here phospholipid molecules. Yes, phospholipids are there. If you take this single phospholipid, so you can see here the hydrophobic head and their hydrophilic tails are forming a core. As the hydrophilic fatty acid tails forming a core, so it is called as a hydrophobic core, right? Hydrophobic core and hydrophilic heads on either side of this lipid bilayer, right? Okay, so it forms a layer like structure, and as this entire, that is hydrophilic head and hydrophobic tails, they are quite flexible, right? They are quite flexible, so that gives a fluidity to the membrane. Exactly like this, the membrane is quasi fluid. We said that the membrane is what? Quasi fluid. So all the molecules, they are in a continuous motion, right? And it has also various kinds of the proteins. We said that integral proteins are there, peripheral proteins are there, glycoproteins are also there, right? And those are integral proteins which involved in transport of the molecule across the membrane. They act as a channel for the transport of the molecules from cell outside to cell inside and from cell inside to cell outside, right? So here is one more example, one more structure is showing that things, right? So just a second. Fine. So in this video you can see that the movement of the molecule across the membrane. Now here is the cell inside, cell outside. The membrane is entirely fluid in nature, and how the molecules, they have been transported across the membrane. You can see here the molecules, they are getting inside, as well as the molecules are also going outside, right, through the membrane. So those are hydrophobic, can pass the layer, that is lipid bilayer. Those are hydrophilic, also get entry through the channels, that is the protein molecules.